All of us have their own favorite organisms for our studies, but not all of us are lucky enough to be able to breed them. In the best case, we're able to go to the field and collect fresh tissue. In our case, Franziska, a PhD student in the priority program TAXON-OMICS, was able to collect moths in the field and preserve legs in absolute ethanol for molecular genetic laboratory analysis. She keeps them in the freezer and can take out comparative samples, such as these larvae, whenever she wants. However, there are reasons she can't travel due to inaccessibility for various reasons. She also had to rely on pinned moths from museum collections and used ancient DNA methodology to analyze them in the lab. The ancient DNA facility of the Museum of Zoology in Senckenberg Dresden is located in a different building than the main DNA laboratory to minimize contamination with intact DNA and especially PCR products. External cleaning companies would bring in more potential contamination, thus we need to clean the facility ourselves. The ancient DNA facility is structured into 2 rooms. The outer one, of cleanliness level gray, and the inner one is kept even cleaner, so we call it level white. The rooms are irradiated with technical UVC light for 4 hours at night. A special dress code is necessary for genetic work on museum and subfossil tissue samples to minimize all kinds of possible contamination with intact DNA and PCR products. In NGS times, this is crucial for later library preparations. To decrease the amount of exogenic DNA, wear a first pair of gloves. To dress in a single-use lab coat, a new mask and a hairnet. Make a hole into the lab coat arms where your thumb should be to make sure they stay in place.
Und zwar mache ich einen Schnitt ungefähr da, wo mein Daumen rauskommen müsste. Mit der sterilen Schere kann man auch einen zweiten machen. So, und dann einfach einmal den Daumen durch. Im Zweifelsfall ein bisschen weiter aufreißen. So, wichtig ist halt, dass der Arm dann nicht wieder runterrutschen kann. Deswegen machen wir das doch und so unseren Arm immer vollständig bedeckt haben, um die Proben nicht zu kontaminieren. Jetzt kommt noch ein zweites Paar Handschuhe. Wichtig, das erste Paar Handschuhe nach dem Anlegen erst dekontaminieren mit DNA-Array oder ähnlichem und dann das zweite Paar Handschuhe überziehen, damit wir hier nicht gleich wieder die zweite Paar Handschuhe kontaminieren und unsere Proben möglichst sauber halten können.
After that, decontaminate or replace the first pair of gloves. Use a second pair to further prevent human contamination. Always work underneath a hood to stop possible contamination from air conditioning and floating particles. The bench and all tools should be cleaned by UV light and DNA away before you use them for your samples. Be sure to clean the tools in between the samples to prevent cross-contamination. We use the NEB Monarch PCR and oligo cleanup kit for DNA extraction. Tissue samples should only be introduced into the facility in a sealed container. Lepidoptera legs can be easily transported in tubes. Always clean tubes containing your samples by using DNA away or similar before introducing them into the hood, as exogenous DNA may stick to the tubes no matter how careful they were handled before. Keep in mind to clean all used tools by UV light and DNA away before you use them for your samples, and keep them clean until usage. This includes all scissors, tweezers, and stainless steel balls in our case. The stainless steel balls are used to crush the legs into fine powder to maximize DNA output in the destructive DNA extraction approach. They are placed into the same tube as the tissue. Remember to clean the tools in between all samples to prevent cross-contamination. For the minimal destructive approach, no shredding of the leg is needed. Place an appropriate amount of lysis buffer into the tube before you transfer the leg to keep statics from interfering with the transition. Insect legs are prone to flip away if not carefully handled. The leg should be mostly covered by the lysis buffer, but don't use too much, as this will force you to use much more of the other chemicals later on as well. Remember to always clean tools after each individual sample and after the last sample too, otherwise you may find sequences of previous projects in your libraries later on. Lysis is carried out at 56 degrees overnight to ensure that all tissue is degraded. In the non-destructive approach, in which the samples should be kept intact after lysis, nevertheless make sure to use enough lysis buffer to keep the leg from breaking.
Für das Typenmaterial, da wir das Bein nicht beschädigen wollen, müssen wir die Geschwindigkeit für diesen Verrückter herunterstellen. Wir haben jetzt noch bei 500, gehen wir auf das Minimale, das ist jetzt 300 bei dieser Maschine, sodass sich wirklich nur noch eine leichte Bewegung erzeugt wird.
For type material, apply only minimal shaking for only a slight movement to prevent mechanical damage. At this point, we change from the grey room to the cleaner white room at the back and make sure to handle samples and lysis products in different hoods, as air-floating scales of previous samples may otherwise lead to contamination. Change or decontaminate your gloves after every sample and/or after every step in the protocol. Liquid-based free-floating scales in the lysis product may clog the silica columns later on, which is why we advise a short spin down of the sample to collect the scales at the bottom of the tube. The Monarch PCR and DNA cleanup kit comes with a specific protocol for oligo cleanup. Use this protocol for the DNA extraction to keep small fragments from being washed out during the next steps. Be careful to use the appropriate volumes of binding buffer and ethanol in comparison to your sample volume, since the protocol uses an initial amount of 50 microliters lysis product from your sample. Then you have to adjust the other volumes accordingly if you are working in a higher volume. The high ratio of ethanol used in this protocol enables the retention of shorter and single-stranded DNA fragments, which make up most of the DNA found in museum specimens. After adding binding buffer and ethanol, make sure to pipette mix thoroughly, at least 10 times with a 100 microliter pipette. The Monarch kit comes with a silica gel column, which is advertised for less liquid retention than other silica columns, allowing us to retrieve most of the remaining DNA from our samples. The column is placed in the collection tube and the sample transferred to the column. While spinning down the sample, the DNA is bound to the silica gel. Make sure to keep a low velocity while doing so, as described in the protocol. Otherwise, you may lose your sample. Spin for 1 minute, then discard the flow-through. Take care not to contaminate the column with remaining drops of flow-through after having discarded this. To wash the DNA bound to the silica gel on the column, use the wash buffer of the kit. Spin down and discard the flow-through. To minimize impurities, wash the sample 2 times by adding wash buffer, spinning, and discarding the flow-through. In case of valuable samples, use a new collection tube after every washing step. For the final elution of the DNA, place the column in a new 1.5 milliliter tube to collect the target DNA. Contrary to the original protocol, we perform the elution in 2 steps of 15 microliters elution buffer each, with an incubation time of 10 minutes for each elution, to maximize the amount of extracted DNA. After you have finally retrieved your DNA extract, clean all hoods you used and tools which you may have touched at any point during your work to minimize possible cross-contamination between projects and samples. Switch on the UV light in the hoods. After finishing the project, also clean the floor of the rooms in which you worked with bleach, chlorine in addition. This will ensure future projects not to be contaminated by previous projects. Finally, before leaving the rooms, switch on the UV light and ensure radiation of 4 hours